Welcome to Bio 344. I'm your instructor, Dr. Robert Bodie, and this is a class on evolution. Now, if you're taking this online, then you are watching these videos, and I'll let you know that these videos are going to very closely replicate what we're going to do in class. The notes will be the exact same, the slides will be the exact same. They'll be a little quicker generally, and they'll have more thumping in the background often. And also, if you're taking the class entirely online, then for the fun fact, you actually don't know if I'm a human or a satyr. I could actually, or satyr, I could actually be pure goat from the waist down because you'll only see me from the waist up. And it doesn't hurt to know that Bodie is Portuguese for goat, so maybe I am half goat, half man. You'll never know if you're taking it only online. Okay, first off, why do we need a class on evolution? It seems like at a Christian university, people will often ask me, how do you teach this at St. Martin's? Well, there is a controversy in this evolutionary theory versus the doctrine of creation. I'm actually relatively embroiled in this controversy and often argue with people about this. So evolutionary theory is a theory, which means it is a series of hypotheses supported by experiments. We acknowledge that microevolution is real. It's something that will happen. There is variation within a species, and species do change over time. We also believe that new species are constantly evolving over the past 4.54 billion years of Earth's existence, and that humans evolved from other apes. Now I say other apes because humans are still apes. We still have binocular vision, opposable thumbs, and then a lengthened period of caring for offspring. Hear the thumping noises upstairs. This is in moderate opposition to the doctrine of creation. Now, it is not a theory. The doctrine of creation is a doctrine, which means people are often required to believe it without testing it. Whereas a theory must be tested, a doctrine really shouldn't be subjected to testing. They believe microevolution is real, that species do change over time. That's the modern version of it. Older versions said that species do not change at all. They believe all species were created in 6 days, exactly 6,026 years ago. Sorry, it's been 3 years since I updated this. And they believe that man was created specially by the very hands and breath of God from clay. So this is a doctrine, though, and there is a controversy between these, especially on how old the earth is and where man comes from. And this does start to matter for people who believe strongly in the doctrine of creation. So if you are of the Christian denomination, you should know that there are many people who say that you aren't of the Christian denomination if you don't believe the Bible. And if you're saying, but I do believe the Bible, well then, do you believe every single word is fundamentally accurate in the way that it is portrayed in our current language? If so, then yes, the Earth is only 6,026 years old, precisely, if you do the math a certain way. And that this is, everything was created in 6 days. Man is not related to apes. You have to believe these things according to a certain group of people. And that group of people is, well, often your fundamentalist Baptists. So there is a controversy here, and some people will say that by taking this class and questioning your faith, you are going to be in danger of the flames of hell. If this is one of those moments where you realize, I'm concerned about learning this stuff, there is the option of not taking this class as well. I'm not going to require in this class that you validate evolution and say, this is what I believe and I don't believe the Bible. No, but I am going to introduce you to a lot of controversy. So if you are uncomfortable, there's the door, and that's fine. Why am I even allowed to teach this? Well, this is a Catholic university, and Pope Pius XII affirmed that evolution doesn't contradict theology so long as God is placed first. So we are, in a Catholic university, allowed to teach evolution. That's fine. There isn't a problem with Catholics teaching evolution any more than there is a problem with Catholics believing that people of different races should marry. That is controversial in other sects of Christianity, but in Catholicism, these are both, well, these are normal things. It's not a problem to teach about evolution from the Catholic view. I'm not a Catholic. I'm a Baptist. In fact, I belong to a fundamental Baptist church. I was educated in creation-only education, where people who would say something so earth-shattering as the Grand Canyon is old they are in very danger of the fires of hell. So I was taught that to teach and learn evolution is fundamentally wrong. The Baptist church that I belong to affirms a 6,000-year-old earth, 6 literal days of creation, one man from whom all are derived. That's Wayne Grudem's Systematic Theology core textbook of Baptists. I don't believe that, and I believe it's a lot much more complicated, and yet I am still able to work this with my faith, and I am even a deacon at Timberline Baptist Church, a local Baptist church where I've been invited to become an elder twice. So it is a problem for some and more of a problem for others. My pastor is a creationist. We have good talks, but I also know people who say that you can't start believing this, and if you do, you're going to hell. So this is kind of a touchy subject, and I am right in the middle of it. So this also lets you know that if you have problems and issues with evolution, it's good to know right off the bat that I've dealt personally with the issues that you're probably dealing with very personally, and that this is a fine thing to come to talk to me about. And I will respect your beliefs, much as I respect the beliefs of other people at my church and other people in science. So it's fine to have a bit of questioning. And yeah. Okay. So this is kind of address the controversy first, and now we're going to get into the fun of it, understanding HIV. We're starting evolution here with an immunocompromised individual and thinking about how that individual got immunocompromised by a virus that should be detected and destroyed by an immune system. So the objectives of today's lecture are to define evolution, selection, variability, HIV, AIDS, and anything else that you're not familiar with yet, you probably want to be able to define. Be able to relate natural selection to AZT resistance. List mechanisms of HIV infection, persistence, and evolution. Now remember, this is microevolution, and since viruses aren't alive, it's barely even that. But let's move in. So HIV, what is this? Well, that's going to be human immunodeficiency virus. It is a virus attacking the human immune system, specifically Homo sapiens. There's also simian immunosusceptibility virus, deficiency virus. We'll talk about that later. So HIV is a global problem. It's also a relatively recent problem, having come out only in the last century. And one of the questions we are being asked is, can this be cured? Can there be an HIV vaccine? Can there be an HIV medicine? Is there a way that we can stop this global plague? And we're talking a global plague with about 3,000 or more people in sub-Saharan Africa dying per day. So that's a massive number of people dying in that area, and we need to think about this as a real problem, even if you do not personally know anyone with HIV. So what is a virus? Well, a virus has everything that is needed to study evolution, even though it itself is not specifically alive. A virus has DNA or RNA instructions. It has a protein coat, which is often used to merge with and invade cells. And viruses are generally very host-specific, meaning they only infect 1 or 2 species. And viruses have 3 things that we can use to study evolution. The first thing is heritability. Heritability is the ability to produce others that are like you. And this is because each virus has DNA or RNA present in each virus particle or virion. So this means that it transmits this blueprint message from one generation to the next, and it is heritable. So any trait it has is heritable. These traits, of course, are variable. The variability exists in all different types of viruses. We are familiar with people getting different types of cold and how bad a summer cold can be compared to a winter cold, or how you catch a cold and then 3 weeks later you catch a different cold, even though they are both the rhinovirus. There is variability in there because often with viruses, mutations are very frequent. It was no surprise that within a few months of coronavirus coming out in large numbers, there were 8 different types of coronavirus. Nor was it a surprise when one of them became more infectious in America, the A something something G mutation. Nor is it a surprise when one of them became more infectious in Great Britain, variability happens, and mutations are frequent, and some of them are going to lead to differential survival and reproduction. If you have viruses that are able to transmit changes in a variable trait down to the next generation, the ones that have the most successful group of traits are going to be the ones who are going to replicate the most and thus are going to survive at a higher rate and reproduce at a higher rate. Now this also has to do with the immune system, where viruses that get recognized by the immune system get destroyed by the immune system and thus will not reproduce. So dead viruses don't reproduce. Can it be dead if it never lived? Well, okay, it's gone. It can't reproduce. So this means differential survival and reproduction is going to mean some are going to exist at a higher frequency than others. And these are the three things of which evolution is made: heritability, variability, and differential survival and reproduction. Without any one of these three, you don't have evolution. A virus, although not alive, is able to have all three of these and thus can evolve. So, how does HIV get inside a host? Before it's going to be able to evolve, it needs to get inside. So let's talk about invasion. We're going to also cover this with the idea of immunity. So HIV will recognize receptors on a host cell. So what happens is this protein coat of the virus is going to recognize the CD4 receptor on a white blood cell, and it will bind to the CD4 receptor, fuse to the cell membrane, and that fusion to the cell membrane is going to allow it to release RNA and reverse transcriptase into the host. Now, reverse transcriptase can turn RNA into DNA. So the RNA will start going into the DNA, will actually be coded into the very genome of the host, and then the host will use that new gene to make proteins, and these proteins are going to be what is needed to make new HIV molecules. These proteins are things like the protein coat, like the reverse transcriptase, like various proteases, spiker proteins, and stuff like that. So all of these proteins are coded for in the DNA of the cell that comes from reverse transcriptase transcribing RNA into DNA. Now, your cell doesn't have reverse transcriptase normally because that's just not a gene we use. So that's a good thing that's different between HIV and humans. How does our immune system function? Well, we have things like helper T cells. These are going to recognize antigens. We have B cells, which are going to be producing antibodies. Now, these antibodies are acting as a selective pressure on viruses. If an antibody recognizes a protein on the outside of the virus, then that virus is not going to survive. If you think back to the blood types, where you have A blood type and B blood type, you'll remember the antigen-antibody reactions between type A and the anti-A that would be present in someone with type B blood. So these antibodies will destroy foreign things, and these foreign things may be HIV. Any HIV virions that are recognized by the antibodies are not going to reproduce, so they act as a selective pressure. Killer T cells are going to look for infected cells, so they are going to recognize certain proteins on the outside of infected cells, and they're going to kill the infected cells. This also acts as a force of selection against HIV. Memory T cells are going to prevent future infections, so when you got a vaccine of a virus, it'll actually it'll allow for certain memory T cells to become more frequent and prevent future infections. So this is how our immune system kind of generally functions with the B cells and the T cells. These are acting to kind of recognize and destroy foreign things, and then remember what these foreign things looked like so they can destroy them if they show up again, which is all going to exert a natural selective pressure against any virus. Now, most viruses are simply going to die out in the body after a series of symptoms, but HIV is a little different. HIV is going to attack the system that tries to destroy it, so it's waiting for an attack here. HIV is going to infect cells with that CD4 receptor, which includes macrophages, effector helper T cells, and memory helper T cells. So it's going to look for these very cells that become more frequent during viral infections. And this can lead to a very long-term infection because not only does HIV attack these cells, but it puts its RNA in the DNA of these cells, thus allowing it to kind of sit latently and produce very few virions at certain times. And that's what we're going to talk about with viral load. So viral load is how many virions are present in an individual at a given time. So how many HIV RNA copies per milliliter of plasma? You take a milliliter of plasma and you see how fast a qPCR is going to act. That's going to tell you how infected someone is. Now you may have heard of qPCR being used in coronavirus testing, because if an individual has a qPCR value lower than 4 replications in qPCR, some people will say, well, they're barely infected at all, they can't spread it. Well, that's a problem for the virus. A virus that exists at a very low level of a viral load is not going to spread very fast. A virus that exists at a very high level of viral load is going to kill the host. Here's the difference here. So you want the host to last a long time and have a chronic, lengthy infection at low viral loads, or do you want the host to, be going out in a blaze of glory, infecting everyone nearby? These are different strategies for viruses, and HIV has both strategies. At first, you'll see an acute infection where there's a high viral load. Then the body is going to react to that high viral load using the B cells and killer T cells and destroy most, but not all, infected cells where the viral load is going to decrease by 3 orders of magnitude or so. And it's going to stay that low level for a chronic period of time up to 7 or more years. And then eventually it's going to increase again. Well, what's causing that? Well, what's causing that is the level of CD4 T cells. At first very high, they get destroyed by that first bout, but they come back and circulate in the blood again. And over time, these CD4 T cells are going to be destroyed by HIV, which infects CD4 T cells. Remember, it binds to the CD4 receptor on T cells and is going to infect them, thus destroying them. Now, if you get below 200 cells per cubic millimeter, then you just have too low of T cells to really respond to infections, which is when an individual is going to get acquired immunodeficiency syndrome, AIDS. That's when the T cell count has basically been exhausted by this chronic and long-term infection. HIV is going to do something to this as well to increase its own survival. So these are some cycles here where the immune system is going to be, so HIV infection will deplete the CD4 T cells in the gut, which is going to end up damaging gut tissues. Impaired gut defenses allow certain bacteria and their products to get into the bloodstream, which triggers an immune reaction. That immune reaction is going to increase the number of B cells and T cells. And more B cells and T cells are basically more sources for HIV to infect. So HIV is going to infect a certain amount of those and replicate prolifically. It's going to be more effective when there's other infections going on. So it actually encourages these infections to exist. When the immune system fights HIV, that actually helps HIV have more food. And over time, this is going to continue to damage the lymph nodes and exhaust where new T cells can come out. So over time, HIV causes more HIV to come out by each infection cycle, and eventually the CD4 T cells are so depleted that the individual can't defend itself against anything. Well, we've got a medicine for this. AZT. So AZT, azithromycin, looks like a nucleotide, and reverse transcriptase is going to use this as a nucleotide. When reverse transcriptase uses azithromycin triphosphate as a nucleotide instead of thymidine triphosphate, it's going to stop. That's going to stop the replication of RNA into DNA. So reverse transcriptase fails right there. Okay, so now we have destroyed reverse transcriptase's ability to incorporate HIV into new CD4 T cells. So new CD4 T cells in the presence of AZT are not going to get infected by HIV because HIV cannot use reverse transcriptase to put its RNA into the DNA. That was a word soup sandwich. Word soup and sandwich, yep. All right, but HIV is going to evolve. Oh no, evolution. That's a problem here. So that's why we came to this class, but this is a problem for the AZT. Over generations of the virus, there's a change in genetic frequencies of how susceptible they are to AZT. What happens is certain types of reverse transcriptase might not bind to AZT and use it. And if they don't bind to AZT and use it, then AZT cannot affect reverse transcriptase's ability to make RNA into DNA. Or it puts the AZT in and then takes the AZT out. And that kind of reverse transcriptase is also relatively immune to AZT. So in the presence of AZT, which HIV virions are going to produce the most offspring? Those that are resistant. So over time, you're going to have natural selection for the ones that are the most resistant. Because if you're partially resistant to AZT, each virion may be able to produce a few more infected cells. But if you're highly resistant to AZT, then each virion can produce even more of these. And you do note that AZT does not take out the HIV gene from existing CD4 T cells, so infected cells will continue to produce virions. AZT doesn't cure, it merely keeps the viral load down for a certain amount of time, which is really unfortunate. It keeps it down until HIV evolves. The population changes over time. So why is it lethal? Well, it infects the immune system and thrives when it's being fought. Eventually, the immune system begins to become exhausted. And that is what's going to lead to AIDS. So, when there's just no more T cells around, it leads to AIDS. And the individual is no longer able to defend itself against tumors, against viral infections, against bacteria, which means the host dies. So in this host versus virus, the virus will eventually win and lose at the same time. What's going to happen is if the virus kills the host, then all virus particles existing in the host die at the same time. This is why some countries that have a very high death rate of coronavirus can say that we're winning the fight against coronavirus because we've killed more coronavirus than any other nation. Every time an individual dies, they take thousands with them. It's because viruses they're not goal-oriented. They don't have the goal of keeping the host alive longer. Now, if they do keep the host alive longer, what happens is they can infect more, but eventually, when they get the viral load too high, the host dies. So it's kind of we know where it's eventually going to go is taking out more virions. This would lead to the hypothesis that viruses that have a longer latency are going to be more effective at spreading. And this is what we see in places like Amsterdam, where we actually have an intermediate viral load, are able to survive, last the longest, and pass themselves on the best. So having a low viral load compared to a high viral load means this individual is, these viruses living in the individual are able to pass themselves on more efficiently. So there is the evolution of viruses that spread more but don't kill more. And that's one way that a virus can evolve, to spread more but not kill more, to become less lethal, more spready. Think the common cold. You walk around with a common cold. You don't put a mask on when you have the common cold. You go out and sneeze with a common cold and get it everywhere because it's not really stopping you. Now think about Ebola. You die, and you don't spread it. Ebola is a poor spreader because it has such a high viral load that it kills the individual. The common cold is such a good spreader because it has a low viral load and keeps the individual alive. Now, a virus can go one way or the other. Things like syphilis moved and evolved towards having a lower viral load. And this is really, HIV doesn't have what's called an escape strategy. So, I kind of already pre-shot this, might come back to it. Evolution of viral load. Okay. Here's what I just said. Rewind and watch again. Remember, HIV doesn't have an escape strategy. Evolution is not goal-oriented. It's got natural selection to evade defenses, natural selection to reproduce faster, natural selection to infect new cells. These things cause a virus to inevitably overwhelm the host and both die. Viruses that don't do this tend to survive better, but, we're going to come back to the but here. Natural selection really is short-sighted. And that is because in a host, the HIV is going to continue to evolve, but the host does not. Strains change within a host. Over the course of 7 to 9 years, you have HIV really evolving, outwitting the host detection ability. Destroying lymph nodes means no new T cells that could recognize the new HIV. So HIV evolves, and the host's ability to detect newly evolved strains decreases over time. And here's the other part, here's the but. HIV evolves to replicate more. And I think between HIV-infected individuals, between populations of HIV, the ones that have a lower viral load are going to be the ones that spread longer. But it's always lethal, because a more competitive strain will always come to rise within an individual, within a population of HIV. The one that spreads the fastest is going to overwhelm all the others and lead to the host death. So eventually, a strain is going to arise within an individual that kills the individual. So that's the goal-oriented, is spread more, but also infect more, and one of those is going to win out. Within an individual, the infect faster is going to eventually win out. And that's something called the HIV X4, the new model. And that's a strain of HIV that can attack naive T cells. This further hastens the decline of the immune system. So new T cells are being infected faster, and the CD4 T cell count goes down way faster in individuals that have the X4 evolution. Okay, evolution. So we've kind of seen a couple different ways where this is not a goal-oriented virus, but what it does is it's going to have changes in its genetics, and these changes in genetics are going to lead to variants that are going to be able to reproduce more rapidly. So we see within a population a descent with modification. HIV at the end of infection is descended from, but looks different than, the original infected virions. We see the change in genetic frequencies over variations. HIV has many generations in a single host, and the genetic frequencies change as the virus adapts to its host's abilities to defend it. Both of the terms in bold are perfectly acceptable for the question, what is evolution? Is it descent with modification? Yes. Is it the change in genetic frequencies over generations? Yes. So these are the two definitions of evolution, and I hope you've seen both in the evolution of HIV. We'll come back to this in another lecture where we're going to discuss the phylogeny of HIV and some evidence for evolution.